So, but don't do this the whole time. So we will share this. Cool. Well, like Helen said, I guess it'll be nice and intimate, just us in this room. No one's in the Zoom, but if anyone hops on, I'm still kind of speaking at the computer too. So yeah. So yeah, a little bit of what we really wanted to do with all of you guys was kind of give you the formal orientation about, you know, all the work that you guys have been doing. What does it feed into? Where does it go? You know, you guys sit and kind of stare through the microscopes and ID all these bugs and help us with that process. But what does that data do for us? Why do we actually look at these bugs? And how does it help all of us in the community understand more about watershed health, stream health, and how to, you know, mitigate future impacts, mitigate things like fire impact, climate change impact, and just kind of keep an eye on overall stream health. So we're going to talk a little bit about who we are, what we do overall at Sierra Streams Institute. So those of you who aren't familiar with everything that we do, we'll give you a brief overview of that. And then a little bit of a deep dive into why your work specifically in the benthic macroinvertebrates matters to us. So first off, I'm Jeff Lauder. I'm the executive director here. I'm a fairly new executive director, just past a year in this position. And we'll talk a little bit about kind of how we came into this building and me as executive director and kind of this new current iteration of Sierra Streams Institute after our pretty lengthy history. So in 1995, we were started as the Friends of Deer Creek. We were originally a friends group, meaning it was a group of concerned citizens got together and said, hey, there's some things going on in this stream that no one's really paying attention to. One of the founders of the group actually noticed some kind of sludge looking runoff coming out of Gold Run Creek right here below the Pine Street Bridge. Reached out to the city, said, hey, what's going on here? And they said, I don't know, it's always looked like that. And they said, well, maybe we should monitor that and keep an eye on what's going on. And it just so happened right around that time, the city had elected to rebuild the Pine Street Bridge, which was a historical bridge connecting the Zion side of town to downtown. And so right around that time, a group of concerned citizens got together and said, hey, can we monitor the stream to look at mining impacts like this sludge or whatever's coming out of Gold Run Creek, as well as make sure that when they do the construction, all of their mitigation measures are actually being followed. We can kind of be a watchdog organization and keep an eye on overall stream health. That then expanded out into things like bioassessment, which is a lot of what you guys are helping with the benthic macroinvertebrate ID. And that was kind of our bread and butter for many years, was just monitoring Deer Creek, which is the creek that runs from up above Scotts Flat all the way down to the confluence with the Yuba near Smartsville. And we focused on primarily stream health in Deer Creek, related to impacts from dams, mining, construction, fire, drought, all those kinds of things, and just kind of doing some baseline monitoring, which we'll talk about in a little bit too, just to keep an eye on general stream conditions. And really the main focus of this was that it was entirely citizen community driven. So this wasn't started by scientists originally, it was started by concerned citizens who said, how can we make sure we're following standardized protocols, doing good science, but we're able to collect data that we can interpret and we can then bring to decision makers. Fast forward to about 2010, 2011, we then reorganized and restructured into Sierra Streams Institute. And that included an expansion of our core programs. So we still maintained the water quality and bioassessment programs, but then expanded out into multiple things under this umbrella of what we call community-based participatory research. And the idea here is, again, it's community-driven. We do research with, for, and sometimes even on community members all throughout Gold Country and throughout the Northern Sierra Nevada. Some current examples of projects we have going on that are under this community-based participatory research umbrella are our monthly water quality monitoring program and bioassessment, which you guys help take part in-stream habitat restoration and native plant restoration projects, health impacts and remediation of mining contaminants. So we have a couple of articles actually about to come out on impacts of mine exposure on breast cancer rates in our region, how to mitigate dust exposure, those kinds of things. Our macroinvertebrate ID lab, which you guys have all been helping out Dave with, and Emma and Helen. And then a lot of forest health research and management plan work. Now a lot of active research and work in the realm of fire mitigation. But all of these things are connected under this umbrella of overall watershed health. And Sierra Streams Institute, before I kind of get into a little bit of what we are and kind of our core identity now, I want to point out, where we were just a couple of years ago. I always like to come back to this photo. This was our office after the Jones fire in 2020. So starting in 1995 all the way through 2020, kind of cruising along, doing the things that we're doing. And then this fire came in a weird way. That was the silver lining of COVID, was that we had some work from home, remote work infrastructure already set up. So it was a little bit easier to kind of still keep work going. But we lost essentially everything. We lost a lot of benthic macroinvertebrate samples that weren't ID'd yet. We lost a little bit of data. Luckily, our data backup process was in place, and we didn't lose all 20 years of our data. We lost just a couple of months. But really, this was kind of a bit of an opportunity to rebuild as well. So we took about a year of kind of still trying to keep active projects rolling while trying to plan what was next for us. Would we rebuild? Would we move? Would we even restructure and change what we did? And we decided to end up in this office. We downsized a bit, focused more on the lab space for the macro lab and the water quality lab. And then the office space is kind of this hybrid space where it's not actually enough space to support our entire staff. We realized through COVID that we don't necessarily need office space for everyone, and that we can continue to do the work that we're doing remotely and in kind of this new hybrid model that we have here now. But we also use it as an opportunity to restructure a bit. So we had a transition in leadership. We had a transition pretty wholesale of a lot of our board members. Dave came in, Lynn Barakos came in, Peter Hollingshead, a student from CSU Sacramento, came in. So we had kind of a wholesale transition of board as well, and then a lot of staff turnover. And so really it was kind of an opportunity to rebuild and kind of envision a new direction for Sierra Streams while maintaining our core programs, which are the benthic macroinvertebrate, bioassessment activities, water quality, and then really just community science is still kind of the driver of everything that we do. So this is a snapshot of our current staff. -1, there's been a bit. Sam has left us, and then now Gary Wells is a new forest technician who's not on here. But all of these staff members fit under the programs listed at the bottom here. We have an education program, rural health, forest health, monitoring and research, restoration and management, and then now we're rolling out our community laboratory services as well to be a water quality lab for the community at large. And so before we take kind of a deep dive into more of the macro stuff, which you guys are all here for, I wanted to give a brief snapshot of our identity now, kind of what we do and where all of these programs fit under this concept of watershed health. So this is kind of the why we do what we do. So watershed health really, in our view, can be summed up as all of these components: forest health, stream health, human health, and then disturbance. So Sierra Streams Institute, we like to sit at kind of the center of these 4 things. Stream health, we assess through bioassessment and through our water quality monitoring, which you guys have all either taken part in or seen some of the sampling that takes place. Human health, we have things like our community health impacts of mine exposure project, mine remediation projects, and focuses on mitigating human impacts of mine exposure. We have a lot of forest health work, both research and actual management for mitigating fire, drought impacts, bark beetle, multiple aspects of forest health. And then we do a lot of research on disturbances as well. So how things like fire and climate change and drought might impact our watershed. But really our programs can kind of be broken down into multiple components of the kind of scientific process that we undergo. And a lot of what we do fits under what we call baseline monitoring. So we can't actually detect changes in the system or an impact from a stress unless we know how that system is just changing on its own over time. So our baseline monitoring programs kind of span all 3 of these main areas: stream health, forest health, and human health, where we're monitoring just current disturbances, current conditions across the landscape and across the watersheds that we work in. We then also utilize natural experiments. This is where nature may apply some sort of treatment that we want to know what the impact of that treatment is. So for example, the river fire was a treatment applied by nature, and we can then look at our water quality data and see what was the impact on phosphate concentrations. Was there retardant showing up in the water? Those kinds of things. And natural experiments allow us to then say what would happen in the future if these conditions start to shift more towards this. We can then also do experiments using treatments as experimental treatments. So for example, an upgrade to the wastewater treatment plant below Lake Wildwood, we were able to see, did that actually work? Did it increase water quality? Through our monthly monitoring, only through baseline monitoring could we detect that change. We then did some salmon gravel restoration work, and we're able to monitor everything from how salmon spawning increased all the way through the whole ecosystem health, like how much bears actually utilize those carcasses, where they moved them, how that moves nitrogen into the landscape. So we can utilize treatments as experiments. And then finally, we can combine all these things into projections. So for example, on the top left is looking at land use change. This is recovery from the 49er fire and how forest type changed over time, how that might have influenced water quality in the lower portion of Deer Creek. Or for example, on the bottom right, this is the Deer Creek watershed, and we're projecting out into 100 years into the future under current climate change models, how might water quality shift at some of these sites, and how can that inform maybe some mitigation or conservation measures that can help kind of mitigate some of those impacts. And then finally, the last step here is all of this, again, fits under the community-based participatory research umbrella, which means that we're interacting with the community through education and two-way communication, where you guys, the community, are helping us collect and analyze data, and then we're sharing that data back with you to tell you what are current conditions, how are they changing, and to see what kinds of questions and concerns you may have based on some of those results. So a lot of everything you guys do is paramount. It's can't even think of the word. It's so important to what we do. Sorry, I still have a little bit of post-COVID brain fog, so I'm not finding all the words I want. But it's literally the essence, the core of everything that we do is that people like you guys coming in, volunteering your time, sitting at those microscopes, looking at those bugs. We can't get the resolution of data that we have, which is frankly incredible, that we have 20 years of data at monthly intervals across this whole watershed and a couple other watersheds. We can't do all of that without you guys. So thank you guys so much. And I'm going to pass it off now to Helen and Emma, and then Dave, who are going to dive a little more specifically into the bugs aspect of what we do.
All right, should we play musical chairs or just go for it?
It's up to you.
Are you even on Zoom?
I am.
Either way. So yeah, so as Jeff said, we have so much data. It's really exciting to be a part of. We have over 2 decades of data. And one of the things that we're doing now, which we won't really get into today, but if you're interested, it's on our YouTube channel. We've been doing a deep dive into the data and how we can, hey, welcome. How we can kind of retarget and reshape our monitoring program in response to what we've found these last 20 years.
I'm just going to put you on the screen for the Zoom, if anyone joined. And for the recording. Oh, let me see. Where's my mouse?
We actually go back. So this is just a map of our monitoring sites. It's actually slightly out of date because we have been doing this kind of reshaping, deciding which sites we need, which sites we can hold off on until we see increases at other sites. But we choose sites based on location in the watershed. We try to get a good spread upper to lower. We look at tributaries, perhaps up and downstream of restoration sites. And then we try to monitor with some frequency. Yeah. So what do we actually do in the creek? A lot of the monitoring that we do throughout the year is of water quality. So like we're actually looking at the water itself and looking at temperature, turbidity, pH, dissolved oxygen, conductivity, nutrients, and bacteria, and trying to get a snapshot of how the creek health is doing at that time. So we try to go out in all different seasons. So like if it's raining really hard, we want to go out then. If it's super hot and low water, we want to go out then. If like it smells weird and we don't know what's going on, we want to go out then. We just want to really like record and get an inventory of the creek in all its different forms. So, but twice a year, we go out and we really get in depth into creek health. And so Emma and I are going to walk you through like the timeline of a benthic macroinvertebrate sample and yeah, talk about the things that we look at and why. So we'll start with Emma because she just spent like 2 months in the field with volunteers collecting a bunch of data. And she's going to tell you about how that.
Does it keep doing? Oh, it's because of the wire. Oh no. Technical difficulties.
Yeah, I feel like those computers have very sensitive tracks.
Yeah.
So we'll just go one path.
Yeah, so this summer, we went out to a bunch of our sites throughout the Deer Creek and Bear River watershed. We aren't able to do full surveys at all of the sites. Some of them are not waitable. For the ones that are, we go out and we will collect water quality, so all the parameters that Helen just mentioned. We will look at the physical habitat, so things like substrate type, depth, the bank, is it stable? Is it vulnerable to erosion? We look at the riparian cover, things of that nature. We will collect algae samples, which we take then back to the lab. And historically, we've analyzed them for biomass, but this year we're going to start doing some chlorophyll analysis as well. We also will do coarse particulate organic matter and fine particulate organic matter, which is also taken back and analyzed for, I guess, biomass. And then the big thing that we go out and collect are our benthic macroinvertebrate samples. So we go out, with a net, and at 11 points along the creek, we go and we'll collect as many of these critters as we can, and we'll bring them back. And that's what you guys get here in the lab. So I'll pass it on over to Helen.
Yes.
Mhm.
So what we get back from the field is this, which doesn't tell you a whole lot. This is kind of meaningless as it is right now. You can tell there's stuff in it, but without the work of the BMI Lab volunteers, this is not data, and this doesn't tell us very much about creek health. So what volunteers in the lab do is to take this, find the bugs, and then identify the bugs. And in doing so, we unlock a bunch of information about creek health. So, I mean, basically, I think everyone here knows, but benthic macroinvertebrate, benthos is the bottom of the water body. Macro means you can see them with the naked eye, and invertebrate means they don't have a backbone. They live in such a variety of creek habitats and have such a variety of tolerance levels to pollution that they act as really good ecosystem health indicators. And they tell us something different than looking at the creek at one point in time. We just look at like dissolved oxygen or temperature. Today at 11:50 a.m. we get a snapshot, but if we look at what can live in the creek, what can survive at this place, we get a much broader scale view of creek health. And so IDing these bugs is really important because it, as I said, unlocks a lot of information about the health of the creek. If we can learn about the tolerance level of bug, the feeding group it fits into, and the habitat it lives in. Go ahead. So I'm going to turn this over to Dave really, but on a broad scale, we use this data to learn more about creek health. We add in all the other data that we collected in the creek, the physical habitat, the water quality, the algae, to learn about this particular stretch of creek. We can compare it to other stretches of creek that we also monitor. And then we can take in the data that other folks have collected, like drought data, climate health data, and just try to get a larger picture of how the ecosystem is doing and how it might change in response to stress. So I'll turn it over to Dave for more information on what we do with the data.
No.
Well, hello. Dave Herbst. I moved here about a year ago and pretty much immediately joined the board of the Sierra Streams Institute because it's very close to my heart, being a stream scientist myself for about the last 40 years. And so have a lot of passion about what Sierra Streams Institute does. So the work that you guys are doing in the lab is really crucial to us being able to generate this kind of data and to have a long-term view of the health of these streams. So here's kind of a view of some of the main players that are there. The one really important thing about bugs as stream health indicators is that there's a lot of them, a really huge variety. There's some of them are relatively tolerant, some of them are relatively sensitive to different conditions of water quality and habitat quality. You'll often hear the term EPT kicked around a lot, which stands for Ephemeroptera, Plecoptera, and Trichoptera, which are the mayflies, stoneflies, and caddisflies. And they're the ones that we really look at as our key indicators the health of the stream. And they're also really important parts of the food web too, because they're usually the larger organisms in the stream. And so they're fed upon by fish and amphibians and riparian birds. So they're really a key part of the food web. They again have these varied tolerances to different kinds of environmental pollution. The density of the organisms that are in the stream is something that we also are able to ascertain because we know what area we sampled out in the field. And after you guys have processed a sample, we know what fraction of the sample you processed. So we know what density of organisms are out there. And that tells us something about how productive the streams are, how much biomass is being produced. And these things change with season, they change with year, they change with different kinds of management strategies going on in streams, and they change with hydrology, with year-to-year variation in the flow. And one of the things that is another important part of what data we collect and are going to expand our collection of is reference sites. So for any kind of water quality sampling, whether it's chemical water quality or biological conditions, you really want to know what's the standard, what's the gold standard against which you can compare whether or not something is in a healthy state. And so having reference streams that are relatively unimpaired by any kind of localized effects from development or land use and so on, allow us to develop that kind of a standard for what we'd expect a healthy stream to look like. So it's key that we have data from those kinds of streams. Do I just tap this to move on? Okay, cool. Oh, and so some of what I prepared in this slide is not showing up, unfortunately, but there should be some arrows and some background on this. But anyway, this just shows you that some of the insects that you saw on that last slide fit into these different sorts of categories for what sorts of food they consume in the stream and the roles they play in the food web. So you have large predatory insects kind of at the top of the food web of these benthic invertebrates. And the kinds of food that other organisms feed on the stream ranges from decomposing leaf and wood litter to algae. And so we have organisms that are grazing on algae in the stream. We have others that are feeding on bits of decomposing leaf and wood material. And as that material breaks up and forms organic food particles, we have other organisms that are filter feeders that pick up that food or that feed on it where it collects in between rocks on the stream bottom. So there's a really complicated food web that develops in streams. And so we do know from the identifications that you guys are doing what kind of a feeding guild or group they fit into. And that tells us a lot about what sorts of energy transfers are going on in the stream and again, how healthy the stream and how rich it is in diversity. Let's see, which one of these?
Just the arrow.
That one, okay, yeah. So let's just hit another little button there. My little animation of key habitats in the stream, and they are riffles and pools. So two of the most fundamental kinds of environments that occur in streams are shallow, swift water environments where there is a lot of oxygenation because currents are turbulent, and they tend to be rocking environments and erosional environments. They're environments where, because of that turbulence, any particulate material that's on the bottom typically is resuspended, is eroded from those spots. And the pools, by contrast, are areas where deposition occurs, where that material that has been pulled up in the current through turbulence reaches a deeper slow water zone, and it falls out of the current and falls onto the bottom of the stream. And so you see these really contrasting kinds of environments from the shallow to deep environments. In the shallow environments, because it's much closer to seeing sunlight, there's a lot more primary productivity going on, which is basically the growth of algae on the stream bottom. Whereas in those deeper areas where deposition goes on, a lot of the organic material, this leaf and wood and fine particles, they settle out and they sit in that environment. So you have really different kinds of food sources that fit into that food web in these different sorts of habitats. So it's a crucial thing also to be able to represent the kinds of habitats that are present in a stream by sampling in both riffle and pool environments. And that's the idea behind what we actually do in the field when we go out with those bee nets and we collect the bugs, is that we're going to different kinds of habitats within the stream, which we're encountering them in proportion to which they occur. So some streams have more riffles, some have more pools. So what we'll see there is representative then of the differences between the riffle and the pool dominating organisms. And so that's this picture gives you an idea of how that distribution changes. So in the center of the slide, you see a deeper blue area. That's a pool area, and so you get these alternating riffles and pools along an undulating current side to side, and as the current undulates deep to shallow, you get this variation in habitats as you go down along the stream. And so in the deeper water portions, you have organisms that are much more tolerant of being able to live in places where there's sediments. So you find a lot of these small organisms, midges, the Chironomidae, which turn out to be organisms that are fairly tolerant of poorer water quality conditions, areas where there's organic matter deposition that's decomposing, also takes a lot of oxygen out of the water. And many of these midges or chironomids are very tolerant to those low oxygen conditions. But there's a variety of other organisms that like to burrow, like certain kinds of dragonfly larvae and alderfly larvae and certain kinds of caddisflies that feed on that decomposing organic matter. Whereas in the shallow areas, you have a lot of filter feeders, because that's where you get a lot of currents and suspended material coming downstream. And so that's the key habitat for them to be able to pick up that suspended material as they filter it out one way or another, often using silk, like a lot of insects are capable of spinning out themselves. And that's also where we see most of the EPT organisms. Most of the mayflies, stoneflies, and caddisflies have a preference for living in those riffle environments. And in fact, there's a much greater diversity of organisms living in the riffle environments. So this is just to say what are the primary players in riffles and pools. And again, my slide formatting didn't work out in this case, unfortunately, but you can see there's differences in the kinds of organisms that occur in riffles versus pools. And a really important thing that we're able to study by having long-term data is what are the consequences of changes in the flow regime. So this gives you sort of a cartoon picture of how streams might change during drought conditions. Those deep blue areas might be areas that represent pools, and then those shallow brownish areas are representative of riffles. And so as streams shrink down because of the lack of flow during drought conditions, you'll see the proportion of those habitats change as well. The riffles being more shallow, or what emerge first as a stream is drying down and the level is falling, and the pools become the more dominant kinds of habitats. And at the extreme kinds of conditions, you'll lose those riffles entirely, and the stream will dry down to a series of intermittent isolated pools. So that has really important consequences on the kind of organisms that you'll find, and the increasing predominance of pool organisms as streams dry down. So our long-term data will help inform what we see in terms of the changes in the environment as a consequence of drought and the changing health and diversity of organisms that are present. So this is another cartoon illustration based on some research that my group did in some streams in the central Sierra Nevada, about 100 miles south here, down near Fresno, maybe a little bit further than that away.
Kings River.
Kings River Experimental Watershed is where this work was done. So under average and high flow conditions, which these pictures are illustrating, you see riffle insects dominating. And so over on the far left there, you see a large predatory stonefly along with a smaller mayfly consumers that are fairly abundant and dominant in the stream. You also see a mayfly grazer that eats algae in the riffles. They're also really important. And then there's another mayfly that inhabits both riffles and pools. The organisms on the left are only riffle dwellers, and the ones on the right are more opportunistic. They can either live in a riffle or a pool. And then as things start to dry down, what you often see is an increase in the overall density. And this is really kind of a concentration effect. So as the levels fall, the organisms that are there are still able to tolerate those falling water level conditions at first and become concentrated because of that smaller volume of water that's there. But as you get into the drought, you see a tremendous decrease in the abundance of the dominant riffle organisms and an increase in the organisms that are able to tolerate both pool and riffle conditions, because even though the riffles are disappearing and the riffle insects as a consequence are as well, some of those organisms that are able to inhabit either environment, they're able to continue to flourish. And so you see winners and losers in this whole game of what happens under drought conditions. But for the most part, it's losers that are occurring as a consequence of those drought conditions. You lose a lot of species diversity. And so as a consequence of losing that species diversity and the density of organisms that are available to the food web, there's going to be knock-on effects. There's going to be cascading effects throughout the rest of the food web. So it will influence whether or not there's food available to fish or riparian birds and bats and spiders. And so these effects in the stream have really important cascading effects throughout food web. So what we study in the streams is really an important key piece of information to how other wildlife may be responding. So just to give you an example of the value of long-term research, I included some data here from studies that I've done that show what things look like. I know there's a lot of noise looking like information right here, but what I want you to see is that on this time scale, it goes from 1998 up to 2020. This is from a study of mine pollution in the Sierra Nevada, not far from here, over at near Monitor Pass. This is Leviathan Mine. And so early on, you see there's a red line here. This is showing you the number of the total species diversity that we find in a number of different stream sites throughout this watershed. And many of the streams that you see represented by individual lines and markers there early on are below these red lines, and that shows that the diversity is very low. On the top is total diversity, on the bottom is the diversity of the mayflies, stoneflies, and caddisflies. But you can see over time that they start to recover. The line that's always on the top that has a filled symbol is the reference site for this particular stream. And so what it demonstrates, bottom line, is that there are a lot of data that we can generate from long-term data sets that allow us to show the success of restoration actions. And so that's basically what this data is demonstrating. It's showing that there's a recovery in most of those streams, all but those that are closest to the mine in this particular case. In most of the species, the EPT taxa, they're slower to recover, again, because they're more sensitive, but they, by and large, in many of those streams have recovered by the evidence from this recent data set. But it also shows other things, like you see a big dip right about 2017 there. And I think I added that as a little, let me see if I put that in there too. Yeah.
No need for this.
And so independent of the effects of acid rain drainage, our data, our long-term data can also show us when things like major hydrologic disturbances change ecosystems. So in spring of 2017, we had this enormous runoff year for a huge amount of snowfall and runoff. This was the year and the time when the Oroville Dam almost overflowed. You all probably remember that. So it has consequences to stream ecosystems as well. And so this data, again, having this long-term data set gives us this interesting insight into the fact that not only are there consequences from pollution, like it occurs with the toxic metals in this mine drainage, but that it can also occur as a consequence of hydrology. But you can also see that in these streams where there's ongoing restoration, the benefits of that accrue almost immediately and rebound. So the effects of things like floods are very short term. Droughts by contrast, like I was showing in that last data set there, have long-term impacts. And so now, more to the point, let's share some of the data from what the Sierra Streams Institute has been collecting over the years. And again, I'm sorry that these somehow didn't format in the same way that maybe it's the difference between putting something into a PowerPoint.
Yeah, Google Slides, and yeah.
Anyway, what we have here is a depiction of how things change downstream. And so the numbers that you see on this graph refer to the numbering system that we have for Deer Creek. So sites 1 and 2 are above the town of Nevada City, and they're fairly high up in the watershed. We consider those to be reference sites because they're least affected by development and urbanization and a lot of other kinds of land uses. Whereas, of course, below Nevada City, there is more influence of urbanization and development, and even more of that as you get below Lake Wildwood, sites 8, 9, and 10. And then site 14 is down near the Yuba River. So the bottom line from this is that we have learned from this long-term monitoring, and this is all including a period of years from 2003 to 2015 is the data set that we have been able to retain. And so in those sites above in the upper area of the Deer Creek watershed, generally speaking, the number of families that are found for the diversity of all the EPT, all the benthic macroinvertebrate families that we found, including insects and non-insects, is in the range of 25 to 30. You get below Nevada City, and it's in the range of 20 to 25, and below Wildwood, 15 to 20. So as you get below these different points of disruption, you see a decrease of about 20 to 25% of what was present in the above reference stream conditions. Now, I don't want to necessarily imply that it's exclusively due to it being below Nevada City or being below Lake Wildwood, although I think that's an important part of the effect. But there's also an elevation gradient here. So we're going from an elevation of probably somewhere around 3,000 feet down to pretty close to sea level at that point, because the site 14 near the Yuba River is at a fairly low elevation. So there could be, an important influence there of elevation as well. But we can tease that apart with further monitoring of other watersheds that don't have the same sort of arrangement of developed areas and undeveloped areas. And then the lower graph is showing us what percent of the total organisms in those samples over time are represented by our sensitive mayfly, stonefly, and caddisfly groups. And so you can see, again, the same kind of pattern above Nevada City versus below Nevada City, higher range above, decreasing range below as you go further downstream, and then below Lake Wildwood, even further loss. Except there's this very odd pattern right there at the end at site 14. Site 14, right near the Yuba, is a site where there's a large amount of gravel, I believe, that was added to try and enhance the habitat for spawning salmon. And so I believe the data that we have from that site, which is relatively recent in time, it doesn't span that whole 2003 to 2015 period. It's actually a relatively short, recent period of time during that gravel enhancement. Well, it looks like there's a lot of EPT in there, but it's important to note that essentially all of them belong to one genus, this mayfly called Betas, which is kind of like the most common insect that we find in streams, that has the really important characteristic of being an excellent colonizer. So it's an insect that in a disturbed area will be one of the first ones that come back, because they do this thing that a lot of insects do, which is called drift. So they'll basically let go of the environment where they're living, and they'll move with the current, and they'll just disperse, presumably because they always have a belief that or an instinct that the grass is greener, right, downstream. So they tend to do a lot of drifting, and there's some other organisms that do that same kind of thing that become important colonizers. And those are also the ones that dominate at this site 14. So I guess the lesson that comes from this is that we need to use different kinds of indicators to make sure we don't misrepresent or misinterpret some of this kind of data.
And it's interesting though, because it does also point out a little bit of like the cause in terms of the impact of Lake Wildwood. The reason we did the gravel augmentation project for salmon spawning was because the dam holds back all the gravel. There's no new gravel recharge coming in for salmon. And so it shows that if you didn't have the dam there and you had more gravel recharge coming in, you might have, more EPT, even if it is Betas. You have more colonization of new gravels.
Yeah, absolutely. And this would be, an interesting place to continue to follow, because the typical pattern is that early on, after a disturbance, like in this case, adding gravel, you'll get all these colonizers, these good colonizers, and a relatively low diversity of organisms. But as time goes past, then other organisms come in that will compete with and displace the ones that are the excellent colonizers, because they tend to be specialists in their capacity to feed and compete and even prey upon these early colonists. So we'd expect to see a change in the diversity. So again, the last sample on this, I think, was from 2015. So to follow up with more current sampling, I think would be really important and interesting to see how these places have responded. So then another piece of data that I thought would be interesting to look at is what kind of trends occur over time. So this is just looking at above versus below town and combining seasons. And so above is blue and below is in that orange brown color. And you can see over time between 2003 and 2015, it's a pretty variable pattern, but always with the sites above having higher diversity of all the BMI families than below town. So we already have seen that in that data that I showed you already. But it also shows us that there is relatively high level of stability within each of those sites, within the sites above versus the sites below. But it also tells us something more about what kind of data would be useful to collect in the future, because again, all the data that we have so far are organisms that have been identified only to the family level. So when we have a higher resolution of identifications, which is the direction we're headed now to identify things to the genus and even to the species level, I think we will see much more consistent patterns and patterns that also may reveal more about how there may be hydrological effects that go on between years when there's different sorts of drought conditions or high flow conditions, for example, like I showed you with that Leviathan mine data.
So you say there's not necessarily a pattern here, but in terms of picking up a little bit of hydrologic impacts, I mean, just considering 2011 and this data set was probably the wettest year of those years, and it seems like there's more separation between above versus below town than in drier years. And then 2012 starts that mega drought. And it seems like as the drought progresses, below town is kind of slightly trending down, but obviously we can't say we would keep doing that.
Right.
We need more data, but
Could be, yeah. So we can speculate and wave our arms with these different kinds of data. But I think the important point is that continuing to collect data long term, improving the resolution of how we identify things, and even comparing different kinds of sites under different kinds of circumstances will be really important. Because one of the problems with this, looking at just this kind of data with the kind of sites we have, is that they neglect the smaller streams where you'd expect more of these hydrological effects to be more pronounced, because they're the ones where, for example, drought is really going to have a substantial influence. So even our higher elevation above town reference tributary streams are relatively large bodies of water. So our site 1, which is above site 2, is right immediately below the confluence of the north and south forks of Deer Creek. And it's really a pretty good sized site. So it's one that you'd expect to have relatively little influence from drought, because it still is going to have persisting high flows in it. It's the smaller tributaries where a lot of this is going to be more evident in terms of these impacts. And in fact, that study that I showed you from the Kings River watershed was done all on small headwater tributary streams to the Kings River. And then I believe I have one more bit of information here. So I think one of the more prominent patterns that we see is the seasonal differences in spring to fall. So this upper graph shows you the richness of, or diversity of, the mayfly, stonefly, caddisfly families at sites in upper Deer Creek. So you see site 1 on the left, site 2 in the middle, and site 4 on the right. And there's a pretty consistent difference between spring and fall, with there being an increased richness. If you were to do a pairwise analysis here for each year, instead of lumping them all like this, you'd see a really consistent pattern that the fall always has a higher diversity, or almost always has a higher diversity than the spring one. So what that tells us is that there's probably recruitment that goes on during the summer, that over the winter months and into the early part of the spring, there's only certain kinds of organisms that can persist and feed on what kinds of foods are available at low temperatures during cold water conditions. But that as the water warms up and as more food becomes available, because there's more sunlight during the summer, you're going to get a lot more algae. And so there's going to be an enhanced recruitment of different species of these mayfly, stonefly, and caddisfly critters. And so you'll see an enhancement as you go from the spring to fall conditions.
Is that pretty standard across other watersheds too, or is that different than other places?
It is pretty standard. Yeah, we see that in a lot of the data from the long-term sampling that we've done at Leviathan Mine, where our reference sites show the same kind of pattern. Yeah. But the other thing we see is that there's a really significant difference in the percent of the habitat that's made up by midges, the percent of the community, I'm sorry, that's made up by midges as you go from spring to fall. There's many more in the spring and many fewer in the fall. And what that probably reflects simply is that there's a lot of emergence that goes on over the spring months, over the summer months, by midges that have developed over winter and early spring months that are primarily feeding on decomposing organic matter. And so as that shifts to algae and as the waters warm up and those larvae are developing into pupae and then into adults, they simply leave the stream. So there's a lower proportion of them as you go from spring to fall conditions. So there's some really consistent patterns that we see seasonally. And so that's a really good thing for us to be continuing to pick up on, are these seasonal patterns, and whether or not these seasonal patterns may shift over time too, perhaps as a consequence of drought. Because that can interfere with the extent to which recruitment can occur in the stream. So the spring samples are really representative of what happens during a high runoff period, whereas the fall samples are really representative of what's happening during this sort of bottleneck period of what we call base flow, which is after all the snow is melted and much of the groundwater has been depleted. And so there's relatively low levels of flow in the streams. So it's really a good design for us to be collecting both in the spring and in the fall. And I think that is it for giving you kind of an overview of what the data look like so far that you and those that came before you have contributed to over the years. So really key, interesting pieces of information, interesting patterns developing, interesting insights to what happens along the course of Deer Creek. I didn't share any of what is going on in Little Deer Creek, where we have a lot of data, or in the Bear River, where we have a fair bit of data. But one of the other things we've started to do this year, in addition to enhancing the taxonomic resolution, so we are identifying things to genus and species level, as we've expanded the geographic scope of where we sample. So we're not just in Deer Creek or the Bear River. We were expanding to tributaries of the South Fork, Middle Fork, and North Fork, and the main stems of those rivers as well. So we have a broader representation of the geography of the watershed as well. And I think that'll really be interesting, because that tells us something about the richness of the diversity that exists in the watershed that I think everyone wants to be able to protect and enjoy and swim in and have bugs to swim with.
And in terms of other, local groups that, do other water quality monitoring, Circle's been monitoring the Yuba for just as long as we've been monitoring Deer Creek. There's Friends of Bear River, Wolf Creek Community Alliance, but we're the only group that does bioassessment. So we're providing this bug information to all these other groups and giving them more insight into what their water quality data does in terms of impact on biological communities. So
are you hoping to have more volunteers and recruit more people to be able to do more?
Absolutely.
Always.
I would say, yeah. I think a key part of this too is for you guys to see not just what data is coming out from the efforts that you put out here in the laboratory, but for you to see, for you to literally be able to come out and see by doing field work as well. So not only do we need assistance with the laboratory work, but also with the field work, because that's becoming, I think, in that we're culling out some of the sampling that we did in Deer Creek and the Bear River. We're adding other new sites that seem like they will be more informative about the kind of data that we collect, but also again expanding this geographic scope. So that means having help in the field as well as in the lab is really key. And I think it gives you more appreciation when you open up a little can of bugs here, your can of worms, so to speak, to see what's in there, to know where it came from, to know the history of data that came from that site, or know what in particular about that site is helping us to answer different kinds of questions about the influence of different sorts of management strategies, restoration practices, hydrological variation from year to year. So being able to come out and experience the site, I think, is both informative for you of what you're doing and a lot of fun.
Yeah, and I know some of you joined or tried to join us in the field this June, July. We'll be going out again in October, but with shorter surveys. We won't be doing physical habitat, which adds on a lot of time. And so it will not be a 9 to 5 gig in October. It'll be more of a morning gig.
Just a 9 to 3. No.
Yeah.
Yeah, Emma and I were just out yesterday, and I think we found that we could do the work in about an hour and a half to 2 hours, collecting the other ancillary habitat data along with the bugs. So it's less of a full day's dedication of time. But if you do have a full day,
you can get a lot
the
done.
physical habitat data is really interesting to collect too, because that tells us a lot more about how we can how well we can understand how the biological patterns conform to the physical environment and the chemical environment that's changing from.